Dear students, as soon as the development of dorsal mesoderm and after that the formation of gastrocele is completed, then the formation of lateral mesoderm begins. And it is completed in two portions. The first portion is known as the somatic mesoderm and the second portion is known as the subplanchnic mesoderm. There is a cavity which is present between these two types of the mesoderm and the cavity is known as the coelom. Now all the cavities are replaced by one cavity known as the coelom. Now after this step, the embryo enters the stage of neurulation. Neurulation is very much important because it is the formation of neural structures which will give rise to the central nervous system. I have given the diagrammatic description side by side as well so that you can understand each and every step. As soon as the cell enters the neurulation stage, the dorsal or the upper surface of the embryo, it has ectoderm, we all know the outer surface, the ectoderm. The ectoderm portion which will become the neural portion is known as neural ectoderm. It is the band of the cells. The cells are large and tough and thick as well. So as soon as the band becomes long, the cells become thick and they form the neural plate. As you can see in the diagram that it will form the neural plate because it will now become hard enough so that you can call it as a plate. And the higher regions, as you can see, they are known as the neural fold. Here you can see the neural fold. Neural fold is present. So with the passage of time, after 21 to 22 hours of the incubation, the neural plate, it folds inside and the folding is longitudinal, due to which there is a formation of groove. Again, a groove or deep structure is formed which is now known as the neural groove. This structure here, this is known as the neural groove. The neural groove edges are called the neural folds. I have already told you about that. This neural groove is visible after 24 hours of incubation. When the 24 hours are completed after incubation, then the neural groove is now clearly visible. Its anterior end forms the brain and the remaining end, as I have already told you, it will form the spinal cord. The neural groove, after both the neural fold, they merge completely with one another, they will give rise to the neural tube. There is the formation of neural tube, and now with the passage of time, this neural tube, it will merge with other layers of the cells present within, and there are certain openings which are present in the neural tube and they are called as neuropores, and these neuropores, they close later after the completion of developmental stages. Now here you can see that the neural tube, it separates from the ectoderm. At this stage, the cavity which is present inside the neural tube is known as neurocele, and this whole process is known as neurulation, in which there is formation of neural tube and its anterior portion will form the brain and the rest posterior portion will form the spinal cord. So dear students, we have completed all the stages of development in the chick embryo, and you have seen every step clearly with the help of diagrams as well. I hope it is now crystal clear to all of you how an embryo undergoes the developmental changes and converts into an adult like its parents. Dear students, you must keep in mind that all the stages of development, they are common with slight similarities and differences and they are common in all the animals, especially the animals which reproduce through sexual method. Dear students, that is all about today's lecture. In the next lecture, we will discuss the various mechanisms by which the developmental procedure is completed, or the changes, the organelles of the cell which control the developmental procedure. They include the gray crescent, the cytoplasm, and the nucleus. We will discuss the role of these organelles one by one in the next lecture. So stay tuned with office.com.pk and keep watching our lectures. Thank you very much for your time and patience. Allah Hafiz.